Good afternoon, everybody, and welcome to today's Research America Alliance member web meeting. As always, we thank you for your partnership and your participation in the Alliance. A couple of quick housekeeping matters. Since we're using Zoom, all audience members will be on mute until time for Q&A, and you can use the electronic raise hand feature if you want to ask a question yourself, or type your question into the Q&A box, and my colleague Terry will pose those to the panelists when they are finished. So today's discussion, I think, is going to be super engaging and interactive as well. It's about the BRAIN Initiative. We're going to hear also about some of the related issues such as neuroethics, and other updates from the National Institute on Neurological Disorders and Stroke. So we're joined by Dr. Walter Koroshetz, the director of NINDS, Dr. John Ngai, the director of the NIH BRAIN Initiative, and Dr. Samantha White, chief of the Scientific and Public Engagement Branch. John and Sam, I think we're going to turn it over to you first to kick things off. Thank you so much for being with us today.
Much, Jenny. So we are really excited to be speaking to the Research America community about the BRAIN Initiative and the NIH's role in the United States BRAIN Initiative. So I'm going to lead us off, and then I'm going to pass the torch to John, who'll then pass the torch to Walter. So for our next slide, I just wanted to give you a very basic overview. So BRAIN is an acronym. It stands for Brain Research through Advancing Innovative Neurotechnologies. And the overarching goal in the United States is to use this initiative to revolutionize our understanding of the human brain by accelerating the development and application of particularly innovative technologies for neuroscience research. So from this, if you could press again, John, there you go. We started the initiative back in April of 2013. And from there, we spent about a year working carefully with the external scientific community at NIH to develop the strategic plan that NIH uses. And we made our first awards for research in the fall of 2014. And we've been working really hard ever since. This is something that we don't do alone. So if you could click once more. We are participating in collaboration with additional federal agencies as well as non-government organizations. So the full list of our partners in the US BRAIN Initiative are at the bottom of this slide. And then we work even more with a few of them on something called the US BRAIN Alliance. So if you go to the next slide, John. With this, we have all come together with the understanding that we each have our own particular mission and are participating in BRAIN with that in mind. So that can be kind of confusing for our public, scientific, and professional communities who stand to participate and benefit in BRAIN. So if you go to braininitiative.org, you can actually have a whole clearinghouse of what is going on all across the country from both the federal and non-governmental perspectives. And we're really working hard to coordinate different funding opportunities, different scientific advancements, news, events that everyone is hosting all across the country in support of the initiative. We also work with this group to create both a Facebook and Twitter presence. So if you're engaged in social media, you can certainly follow us at US BRAIN Alliance or find us just as BRAIN Initiative on Facebook. And that's also another way that we really work hard to expand our outreach for the initiative. So from here, if you are interested in participating in the BRAIN Alliance or learning more, I do encourage you to reach out to me, and Research America is able to provide you with my email for that, because we're really working to align with anyone who's invested in neuroscience research. Now I'm going to pass the torch to our BRAIN Initiative Director, John, who's going to give you a bit more of an in-depth look at how NIH participates in this fantastic effort.
Thanks very much, Sam. That was a great introduction to today's discussion. Again, the NIH is just one of several partners participating in the US BRAIN Initiative. At the NIH, we have 10 participating institutes and centers. And our goal is to develop and apply new tools for understanding how neural circuits underlie complex behaviors, both in the healthy and normal brain, but as well in disease. And to achieve this goal, I think it's important for us to first of all, to leverage the technologies that are coming about to enable new discoveries about neural circuit function. It will be very important to use these discoveries as a foundation for new therapies for human brain disorders. And very importantly, we must disseminate and democratize technologies for basic discovery, as well as for these clinical applications. Now, as Sam mentioned, we started the NIH Brain Initiative, or the Brain Initiative, in 2014. And I think it's fair to say that the success we've enjoyed so far can be attributed in large part to a thoughtful and visionary strategic plan that was commissioned by NIH Director Francis Collins back in 2013. The report was released in 2014. And this was written by a group that was co-chaired by Dr. Corey Bargman at Rockefeller University and Dr. Bill Newsom at Stanford University. And among the many ideas put forward, the group, through the strategic plan called the Brain 2025 report, encouraged us to focus on 7 main scientific areas that overlap. The first is to discover the diversity of cell types in the brain. In other words, to understand what the parts list is of this remarkable biological computer. The second is to develop tools for mapping out the circuit diagrams, the wiring diagrams of the brain at multiple scales. The third is to continue developing technologies to monitor neural activity. And the fourth is to devise tools for intervention, in other words, for modulation, which experimentally can be used to demonstrate causality and therapeutically can be used to treat certain human brain disorders. And then finally, in the last set of approaches, this is really to integrate the knowledge we gain from these tools, grounded in theory, to understand how neural circuits, again, drive behavior. So this is in 2014. We've been going at it for about 5 or 6 years. I'm going to highlight these 3 areas of brain cell types, circuit diagrams, and interventional tools in a moment. But Dr. Collins also realized 5 years in, it would be a good time to reflect on our progress so far and to ask whether we should in any way adjust our plan for the next 5 years. And so he commissioned another working group, the so-called Brain Working Group 2.0. This was a group chaired by Dr. Catherine Dulac at Harvard University and Dr. John Mansell at the University of Chicago. And they wrote a very nice report that looked at our progress and made suggestions for the futures. And their report was entitled From Cells to Circuits Toward Cures. And I'll emphasize the toward cures part at the very end. They had 3 major findings they wanted to leave us with. First of all, they lauded the progress made so far, and they encouraged us to stay on this productive path that was already forged, and to continue support for technology development and the targeted study of circuit components. The second was to be mindful about balancing individual investigator research with team science. So many of the successes we have in brain have been done with large teams. I think certain kinds of data, certain kinds of problems can really only be done at a scaled level. But still, we need to be mindful that we need to have good questions to ask. And here is, I think, really the beauty of the NIH ethos, which is to strongly support individual investigator research, hypothesis-driven research, the research that starts with a good idea that can be solved with better and better tools. And the third point is what I'll spend the remainder of my time on, was a suggestion to reflect on our progress made to date and ask whether over the next 5 years we could start some projects that would truly transform the way we can study the nervous system and ultimately translate those discoveries into new cures. So I'll focus for the rest of the time on these so-called Brain 2.0 transformative projects. And again, they build on a really solid foundation of discovery and technology that's come about over the last 5 years, not only within brain, but in the field of neuroscience and other fields of science more generally. The first project I'll mention is this phase 3 brain cell census project, or the so-called parts list of the brain. The brain cell census project has been going on since 2014. This will be the 3rd phase we're proposing. It will build on the success of efforts carried out so far on characterizing the mouse brain in the so-called Brain Initiative Cell Census Network, or BICCN. And now, starting in 2022 or 2023, we will shift emphasis to construct a comprehensive human brain cell atlas, so a parts list, if you will, of the human brain. The second will dovetail with this effort, and that is to develop new technologies for studying and revealing brain microconnectivity, or the so-called wiring diagram. So if we think of the brain cell census as the part list, the brain microconnectivity analysis will be the wiring diagram. And together, these two pieces, or these two atlases, will really give us some ground truth information about the nature of neural circuits and allow us to ask questions about how they drive behavior at unprecedented levels of detail, and also allow us to identify circuits that are relevant in certain disease states to give us some insights for interventions. The third one is what we call the so-called cell type specific armamentarium. This is a project to organize resources to gain access to cell types in the brain across multiple species. Now, many of us are quite familiar with the genetic tools we have at hand to access cell types in organisms such as the worm, the fly, and the mouse. Ultimately, we're interested in working our way to bigger brains and to understand how neural circuits function in so-called non-genetic organisms. And so we will be developing strategies for gaining access to cell types in the brains in postnatal animals, somatic cell access, using both viral and non-viral mediated technologies. The long-term goal is to develop new therapeutic strategies for human brain disorders along the lines of molecular and gene therapies. And we're very pleased to let you know that the first RFA, request for application, has just been issued with a receipt date in early 2021. So what I'd like to do just in the next few minutes is just to give you some highlights of the foundation that we believe will really propel these three projects. And I'll start with the cell census project. So why do we need a cell census? Why do we need an atlas of cell types? Well, simply put, if we want to understand how this computer works, we need to have a parts list. We need to understand who are the parts on the circuit board, how many of them are there, what are their properties, and very importantly, how are they connected to each other to take in information and process it and send it back out to drive some kind of output or behavior. Now, this is a very old problem dating over 130 years ago, since the beautiful studies of Ramon y Cajal, who described the multitude of cell types in various nervous systems based on morphology and location, but really we need a systematic way to identify and classify them. So this is the basis of the Brain Initiative Cell Census Project. And the idea is to leverage multiple modalities to fully characterize the cell types in mammalian brains. This started as an effort, a pilot effort in the Brain Initiative Cell Census Consortium from 2014 through 2017. We're currently in the second phase, the Brain Initiative Cell Census Network, which will run through 2022. So I just joined the NIH early this spring. Until then, I was a PI on both of these projects, very proud to be associated with this terrific group of scientists. And the idea here is that we'll use molecular signatures based primarily on single cell sequencing technologies as a foundation for understanding different cell types, anatomy, morphology, connectivity, as well as function. And this is an ongoing effort. I don't have time to tell you all the glorious data we're deriving from this project, but it's going quite well, and we're about halfway through the BICCN, and the papers are just starting to roll out. So this has been a large coordinated effort, quite a juggernaut, if you will. It's centered around these so-called large U19 grants, production grants, that are generating and analyzing data from the mouse brain, as well as a series of smaller but quite significant UO1 studies that are focused on understanding connectivity of the cell types that are being discovered. So these are the major mouse projects. But we also have started exploring the capabilities of performing these analyses in big brains. So in humans, as well as in non-human primates. We're also supporting a series of grants to develop better technologies so that we can derive more data for less money and time, and also understand better how to analyze and integrate the data. These efforts are all being coordinated through the Brain Cell Data Center, this U24 grant, which then coordinates all these efforts here and ensures that the data are appropriately deposited in these data archives. Now, data use and reuse are very important for us. And so we've really, I think, done all that we can to step up these data archives and also now more recently data analysis centers to make the data that we are generating from this large project both available but also useful to the entire scientific community. So just to summarize, the idea here is to build a comprehensive brain cell atlas based on multiple modalities. It's very much based in transcriptomic and epigenomic approaches, but it doesn't end there. It involves integrating information about individual cell types with physiology, morphology, and connectivity to give us data-driven cell taxonomies. At the same time, by understanding the genes and enhancers that are being used in these cell types, we can then develop cell targeting tools that will further our efforts toward mapping out connectivity and ultimately function of these different cell types and their connections as they drive specific behaviors. So this is the basis for the cell census project. And again, starting in late 2022 or 23, we expect to step up the third phase of this project that will focus primarily, but not exclusively, on building a comprehensive human brain cell atlas. So next is this project to map brain activity and connectivity through a more comprehensive map of connectivity in the brain. Over the last 5-10 years, we've seen the development of a remarkable array of new actuators and sensors of cellular activity, many of these genetic reporters. We've concurrently developed powerful imaging modalities to visualize activity within the nervous system that takes advantage of these tools. And this has allowed us to start getting at some of the principles underlying the circuit basis of behavior. Now, what we really need, again, is a more fine-grained connectivity map. Again, this will provide a ground truth for circuit analysis and constrain our hypotheses and models going forward. These maps will need to span multiple scales. So while we know that local circuitry is certainly important, recent research over the last few years has also shown that certain behaviors engage different regions of the brain that are quite dispersed geographically, but yet they're connected. So we really do need to understand how different regions of the brain are connected and how they're engaged to drive certain behaviors. We need both a dynamic and a static map. So we not only need what the roadmap looks like, but we also need to understand the traffic patterns. And we need to understand what cell types are participating in these circuits and how they're being engaged during different behaviors, so we can have a better understanding of the principles, the underlying principles underlying coding, but also have a better understanding of what cells to target in different human brain disorders. So let me just show you an example of why I think this is going to be so important for us. Here's an example in the larval zebrafish, and this is from a study conducted by Dr. Florian Engert's group with brain support at Harvard University. And here they're leveraging the power of the larval zebrafish because this allows them to provide a visual stimulus. You can provide other stimuli, observe a behavior. So on the left side, they're showing these fish a screen with flashing dots that are moving in some direction. And the fish is deciding to swim left or right. And they can assay this by which way the tail flicks and with what kind of frequency. They can simultaneously monitor cellular activity in the entire brain more or less simultaneously. So this allows them to understand what the flow of information is through neural circuits. So this is shown in the middle. And again, this is due to the properties of the larval zebrafish in that it is optically transparent and at the larval stage can show these very interesting behaviors. And using a combination of giving different stimuli, observing behavior, and measuring activity across these circuits, they have proposed a circuit motif for decision-making in the hindbrain of the fish, comprising these sets of neurons. And right now, the challenge will be to test these hypotheses and to really understand how they drive the behavior, how perturbations in the circuit affect those behavior, and so on. So I think this really stands as a great example for a motivation for why understanding the connectivity between these cells is so important. Here's a paper that by now is about 5 years old, but it's seminal from Dr. Jeff Lichtman's group at Harvard. And here they've reconstructed at a very fine scale, the nanometer scale, the connections between cells in about a cubic millimeter of mouse cortex using really cutting edge or bleeding edge scanning electron microscopy combined with very computationally intensive reconstruction. And so here is just a rendering of this. Now this is from about a cubic millimeter or so of mouse cortex. The issue is if we want to do this for an entire mouse brain, it's a problem that is several 2 to 3 orders of magnitude larger. And I think the computational challenges will be even larger. So the big challenge that we face is how to scale this up to an entire mammalian brain. And here they've color-coded the different kinds of cells that they're seeing here and the connections. I'll just let this run out. Axons, glia, and other cells, dendrites, these are the processes, and so forth. So this will be a big challenge, and this is what we'll be interested in investing in order to do it. We don't quite have the technology to do this today at a reasonable scale, but I think if we put the challenge out there, I'm sure the field will respond accordingly. Okay, so the third large project we will be starting is the cell type specific armamentarium. And again, this is to develop technologies for enabling both experimental and ultimately therapeutic access to specific cell types in the brain. I just want to give you one example of where this might go based on some really cool molecular biology. So this is from a recent paper from the laboratory of Dr. Viviana Gradinaru at Caltech. And Dr. Gradinaru and colleagues have used directed evolution to select for viral capsids that confer cell type specific tropism. So here she's using, in essence, directed evolution to target viral vectors to different cell types in the brain or in the body. And in this latest method that they've developed called mCreate, it's quite scalable, and they can really select for tropism both for and against different cell types. So here in the schematic, what they've done is they've generated a library from which they can select different viral vectors that can target, first of all, the blood-brain barrier and actually allow access into the brain by finding viral capsids that can actually cross the blood-brain barrier. So this makes the delivery part of the equation quite a bit simpler. And then you can further select for targeting to different cell types in the brain. And very importantly, they can also select away from targeting to other organs, such as liver, kidney, and heart. Now, one of the challenges of gene therapy these days, using adeno-associated virus, or AAV, which right now is the vector of choice, is that there is some toxicity associated with AAV winding up in the liver, kidney, or heart. And by selecting away from these organs, we can actually establish more useful tools, not only for research, but eventually for therapeutic applications. So they've done this selection process, this directed evolution process. They found various capsids that can target AAVs carrying GFP reporters to a wide variety of cell types in the brain. And so this will really serve as a promising platform for tuning these gene delivery vectors. And then combined with the information we're deriving from the cell census projects that are identifying both promoters and enhancers for the specific cell types, I think together these technologies can give exquisite targeting for both research as well as for therapeutic applications. So I just want to finish up by talking about the cures aspect. Ultimately, I think our mission at the NIH is to find and deliver improvements for human health. I think the discoveries supported by BRAIN really do lay a foundation for developing new interventions for human brain disorders. By focusing on unraveling the circuits and computations underlying behaviors using these new tools, I think we really are putting ourselves in a great position to identify the cells and circuits affected in neurologic and neuropsychiatric disorders. This has already led to the development of some quite sophisticated closed loop deep brain stimulation devices. Our blue sky dream here is to develop targeted molecular and gene therapies based on, I expect, the transformative projects that we are starting right now. And already we're seeing some really great applications of the technologies in developing sensory and motor neural prostheses. So I just want to take a moment in my last slide to give you an example of what can be done. And this is, I think, a really impressive example, but also for me, is quite inspiring to imagine the future based on this. So here's a study from Dr. Eddie Chang, a neurosurgeon at UCSF, who's been a pioneer in understanding the neural basis for human speech. And one of Dr. Chang's interests is to use this information to develop neural prostheses for patients who are either paralyzed because of some traumatic event, or otherwise perhaps locked in because of a degenerative disease such as ALS. So what he's done is he's performed interoperative recording with these surface electrodes, ECOG arrays, electrocorticography arrays, in patients that will be undergoing surgery. And through a series of studies, Dr. Chang and colleagues have identified the patterns of neural activity that are associated with speech. And they've also linked this in separate studies to the kinematics that are involved in generating various speech waveforms. So by using these approaches and a recurrent two-stage recurrent neural network, they can record from the brains of patients or subjects as they are either speaking or thinking of speech. Using information that they've derived to infer kinematics, they can train the first stage of this recurrent neural network to establish a series of kinematic outputs here that are again combined with the actual speech waveforms to synthesize a speech pattern based on this. And they've actually done a remarkable job. They can now take speech, recorded electrical activity and generate speech in sentences of over 200 words with a fairly low error rate. So let me just play this for you to show you what this actually looks like. So here is just a depiction of the ECOG array on the surface of a patient's brain. They're now decoding the activity as the patient is either speaking or thinking of speech.
The proof that you are seeking is not available in books.
The proof that you are seeking is not available in books.
The proof that you are seeking is not available in books.
So I think you will agree this is really quite a remarkable accomplishment today, based on really some beautiful neurophysiology in humans, a very challenging effort in and of itself, but really a brilliant application of machine learning algorithms to deconvolve the patterns of activity that are involved in generating this motor behavior. And I like to use this as an example of imagining the impossible, because 5 years ago, this really did seem to be quite impossible. And so I'll use this as a lead into Dr. Koroshetz's comments on the implications of the work that we do on humans and the ethical concerns that we really must be paying attention to make sure that we're doing this for the benefit of all. So Walter?
Thank you, John. Thank you, Sam, and all the people who worked on this amazing project. I did want to spend a little time getting people to think about something I think is very important coming out of the Brain Initiative, and that is an imperative for society, for each of us, to begin thinking about the appropriate use of these technologies as they come along. So the point that's different about the Brain Initiative is, as John said, it is laying out the ability to map, monitor, and modulate brain activity, brain circuit activity, and that's what gives rise to consciousness, our innermost thoughts, and the tools to interrogate and modify brain circuits are going to bring new questions to society. Some of these will be in the medical space, and we have certainly lots of precedents for moving technologies appropriately to patients to improve their functional abilities. But these tools that we're talking about may also, and almost likely will, be adopted outside the medical space. And that brings really new questions to society. So there's a real critical need as we move forward to have an ongoing dialogue between neuroscientists, neuroethicists, policymakers, and people like yourselves and society as a whole. And we need to start thinking about what kind of guiding principles should serve to inform this kind of dialogue about the ethical conduct of the Brain Initiative research as it is, but also how these technologies will be used in the future. So, next slide. So the next slide, I want to apologize, is not any insinuating anything about the ethics, but I want to just make the point that these tools have not escaped the surveillance of entrepreneurs out there. And so the best example is Neuralink, run by Elon Musk, and they have taken some of the brain initiative technologies, improved them, and have amazing ways of now recording from thousands of neurons in a, this is a rodent, but also in a pig they've shown. And the plan is to allow these technologies to run computers and allow people's brains to interface with computers, particularly in the beginning, in people who have severe disabilities, such as those with amyotrophic lateral sclerosis and total paralysis or brainstem stroke. But that's clearly not the end of the company's interest in these technologies that will link people up to computers. And so let's just start thinking about this now. Is we're not too early. Next slide. And so in the Brain Initiative, what we've done at NIH is to try and integrate the neuroethics into the science from day one. So we have a advisory group to the NIH called the Neuroethics Working Group. They put together some neuroethics guiding principles for the initiative, which I'll talk about. They've held workshops about particular areas of concern or interest. And we also fund ethics research, neuroethics research, as part of the Brain Initiative. And also, as John mentioned in the Brain 2.0 report to Dr. Collins, there was a report that came back that was specifically focused on neuroethics in the Brain Initiative. So I guess the good news is that we certainly have taken this incredibly seriously, and we're glad we have right from the beginning. Next slide. This is the last slide. I just wanted to talk to you about some of the guiding principles that have come out of the Neuroethics Working Group for the Brain Initiative. And the first one is obvious, makes assessing safety paramount. So as we do apply these technologies to humans with different disorders, safety is really critical. And then to anticipate the special issue related to capacity, autonomy, and agency, particularly in devices that are going to change neural circuits. So we actually have examples of those, deep brain stimulation for Parkinson's, but now this ability to modulate brain activity is going to go to a completely different level. And many other disorders may be helped by devices or other interventions that change circuits. So who's in charge of that change? Well, who's responsible for behaviors that occur in that spectrum of behaviors that could occur after someone is treated with these type of devices that actually affect their brain activity. The privacy and confidentiality of neural data is certainly easy to understand. John showed you the kind of data that could come out of a brain recording that allows the interpretation of speech before someone actually even makes the attempt to speak. So being able to decode what people are thinking is not something that is that far off in these particular instances. And so privacy and confidentiality of the neural data, and what kind of neural data would you really want to collect, and what would the confines to that be? And then to predict and plan for possible malign uses of neuroscience tools and technologies. This is particularly in the area of enhancement. So we as the National Institute of Health are using these tools to help people who are unable to use their brains as they used to achieve new functional levels. But one can certainly think about applying these to other areas to enhance a normal person's ability to do something. And some of those things may not be so good. And what is good is something that a society is going to have to discuss. And this is, again, really important as we move these tools into both the medical and the non-medical uses. And what we're talking about today is a chance for this dialogue to start and to begin to address specific concerns of the public and get the public thinking about these issues and encourage public education and dialogue. And finally, the last one is to behave justly. And that's, do the right thing and make sure that the benefits of this research and the technologies are accessible to all the people who need them. And so those are the guiding principles that the group has put together. Next slide. And so it'll really be interested in people thinking about these things over the next couple of years or even decades as these tools come out. And I just want to end up by saying the BRAIN Initiative at the NIH level has just been a fantastic, tremendous effort by lots and lots of people who are listed here. So thank you very much and appreciate your attention, and happy to answer questions with John and Sam.
Terrific. Well, I'm sure we have plenty of questions. Thank you, Walter, John, and Sam. I'm going to use my moderator's prerogative and just ask the first question, which is that the BRAIN Initiative just seems to be a really terrific example of research which cuts across NIH as well as federal agencies. I know you partner closely with DARPA, FDA, and NSF. How does this approach, this cross-institute, cross-agency approach, how does it help to stimulate your work?
I'll start. I mean, I think as you mentioned, it's really an opportunity to engage scientists and as well as agencies from across the spectrum of expertise and missions. And so I think this is what attracted me to BRAIN from the very beginning. This is why I was just delighted to have the honor to be a part of this at NIH. We are trying to build these partnerships. There are great opportunities, but also some challenges. There are a lot of complementarity in the different agencies' missions. And many of the investigators that are funded by the different agencies, NIH, NSF, IARPA, DARPA, and so forth, are common to the investigator pool. But they're being asked to apply their resources and know-how in somewhat different ways. And that's what makes this all kind of very interesting. In fact, some studies have started out with funding in one agency and have wound up receiving follow-on or associated funding on offshoot projects through another agency. And I think this is just it's just building a really great ecosystem for the U.S. BRAIN Initiative. And we're looking forward to building that more robustly and also looking outward toward international collaborations. So,
Thanks, John. Terry, I think I'll turn to you. I think we've got some questions from our participants.
We sure do. And just a reminder to folks, they can put their questions in the Q&A box. First, we have a question that may be a niche question, but I think everyone relates to this. We are all patients. This year, this person writes, I had a ganglioglioma on the front part of my left side temporal lobe. I'm curious if there are any researchers or studies where the impact of its removal or how my brain is adjusting to it would be useful for their work. We figure it's not a common situation. So if this health issue could help build on the understanding of brain circuiting, this person would be interested in learning more.
Sorry,
Walter, you just muted yourself. There you go.
Projects in the BRAIN Initiative that are looking at temporal lobe function, particularly in regards to memory. And so some of the really amazing work comes out of the neurosurgical sphere, where when people can undergo this surgery and they volunteer for brain recordings to go on for a couple of minutes, either before or after, we learn tremendous insights into how the human brain is wired around these circuits. So that's really where the action is right now. And I think Dr. Chang's example that John presented is a good one. So thanks for that question.
Terry, others?
Oh, yes, definitely. Are there plans regarding neuroethics? Are there plans for ecosystem-wide neuroethics guidelines? And would this require broader consensus from the research community on which methods are of concern?
Yes, so I think that's a great question, and this is an ongoing effort, so that in the BRAIN Initiative, I'd say that there are issues that have come up that were totally unpredictable. So I think it's got to be a continuing dialogue among the scientists that is focused on the big picture, but also addresses the issues, the particular issues as they present themselves. And then I would also add, John mentioned that the Brain Initiative has a lot of international collaborations going on. Many countries have initiated their own brain initiatives, and so the ethical issues have been one of the things that come up in these international collaborations as well. So that discussion is going on a global effort, which is important because different countries have different cultural norms, and yet we need to, at least discuss the issues that are common to all our different cultures as we move these technologies out. So that's happening, what's called the Brain Initiative Alliance, that is a global effort now.
Right, just to follow on what Walter said, and also what Walter mentioned in his presentation, there are potential pitfalls that come along with these enormous opportunities, and the approach that was employed from the very beginning of BRAIN was to be proactive and anticipatory of what we might be getting into. So literally, I say we don't want to get in over our heads too fast, too soon. And so we have structures in place. We have a neuroethics working group that is, charged with not only making sure that what we're doing is acceptable, but also to anticipate where we might be going based on the research that we're funding. So they're actively engaged in all that we do. We have 8 scientific project teams that are working for the Brain Initiative at NIH. One of those is charged specifically with issues pertaining to neuroethics. Now, they not only assist in the review at the program level of applications and awards that are going in and out the door, but we're also, as Walter mentioned, we're funding research in the space of neuroethics. So this is a scholarly pursuit. This is not just something we need to patch up as we go along. There's a lot of great science to be learned here, a lot of great research that we can use in an evidence-based way to make sure that we're developing and using these technologies for the most possible good.
Great.
Terry?
Another question regarding the cell type census. Are similar cells in similar regions essentially the same across species? And how might the Brain Initiative convey those similarities and differences between models and humans to ensure effective translational research in this area?
Well, that's a great question. If you have a couple hours, I'd be happy to sit down and tell you about it. Part of the current work in the cell census network actually is performing comparative analysis using primarily single cell sequencing, but also integrated approaches that include physiology as well as some connectivity. What we're finding is really quite remarkable. If you look at the size and complexity of a mouse brain, it's about 80 million neurons. The human brain is about 80 billion. So many, many orders of magnitude difference in size and number of cells. And yet when we look at the cell types that we're discovering between mouse, non-human primate, and human brains, the take-home message is by and large, the cell types are conserved. But there's some really interesting exceptions. There are some cell types that are specific to the primate lineage, so non-human primates and humans. But those are the exceptions. And also what we're finding is that the cell type specific markers, so we might see common cell types across all species, but the genes that actually highlight them as being different are different. So I think one of the really interesting and useful take-home messages from this is not only is there evolutionary conservation, but it's there's a lot of really interesting things going on in the differences. But to the point about translation, there's been a lot of hand-wringing about whether mice are a good model for human disease. And I would argue that it is, as long as you know what you're looking at. And so what these kinds of studies tell us is that there will be some cases where certain predictions made in one organism are not going to hold up in another, but we actually now have a roadmap for which ones might be more promising than others. So it's really going to be a great coming set of years as we really start deploying this information. Again, one of our goals is to identify disease-relevant circuits in humans, and armed with this information, I think we'll have a much better map for how to start from these model organisms and get ourselves in a meaningful way, in a successful way, into human brain disorders. So stay tuned. The papers are just starting to appear in bioRxiv, and we're hoping to see these studies in press by sometime in 2021.
I think we have time for 2 more questions.
Okay, another question broadly about patients. How have patients and families been incorporated into brain initiative discussions, planning, and policy formation?
Right, so that's a good question. And what we have tried to do is to encourage, patient engagement at the site level, where patients are enrolled in studies, and for instance, at the brain level, we've looked into a problem which cuts across a lot of sites, which is the fact that in people who volunteer to have interventions done on, for instance, devices placed in the brain, we're trying to work out processes that enable those to be maintained over many years, even after the studies are over. And those kind of discussions include the patients, the payers, the IRBs, and the scientists, as well as our neuroethics group. But these studies that are being done in people at this point are small numbers of people, but they're big commitments on the part of the people who take part in them. And so it's every one of those discussions is on an individual level as well as on a class level.
All right. Terry, do we have another one, or shall I take the last one?
I think you can take the last one.
Okay. So this was actually shared by a colleague. How do we promote equitable access to the knowledge stemming from the Brain Initiative? Is there a way to incorporate health equity into the process? Has that already been built in to, for example, the work that you're doing on the ethical side, or in terms of how you're making your resources available?
Yeah, that's a great question. Thank you for posing that. I feel that if we don't democratize the technologies we develop, both in terms of research tools, but really very importantly in terms of therapeutics, then we will in many ways have failed. I think too much of the biomedical enterprise has been to the benefit of a few and not to all. This is something we are actively engaged in. Recent events around the country, if not the world, have really put a spotlight on a problem that's been with us for quite some time. And we are committed to making sure that we do truly make what we develop accessible to all. This will have to happen at many different levels. It's an issue of who we study, who we choose to put in our study cohorts. This is a particularly intense conversation we're having now as we are planning on moving our studies more squarely into the human domain. So we have to be very mindful of, if we really want to develop tools and molecular and genetic tools for curing human brain disorders, then we must take into account the diversity of humans that are on this planet. That's first of all. We're also recognizing that who we study is very much impacted by who's doing the studying. So by choosing to include this group or that group in a specific scientific endeavor, you're also de facto choosing who to exclude. And somebody's doing that choosing. So these are issues of intense discussion. I think it's, in a way, a timely discussion, especially as we develop tools that I think really will transform our ability to treat human brain disorders. And we welcome engagement from the public on this and from scientific societies. It's a tough problem, but it's one we have to face, both for societal reasons, but also I believe that as a biologist, I've come to appreciate that diversity actually can teach us a lot about how processes work. So I think for both scientific reasons as well as for social reasons, we really do need to pay attention to this. And I've made a very firm commitment to this as part of the mission of the BRAIN Initiative.
Thank you, John. That was a helpful answer to a really important question. Well, I think our time is over with you, Walter, John, and Sam. It's been a stimulating discussion. And I know I've learned a lot, and I think we all have. For those on the call, we'll be posting their slides as well as the video of this conversation. So you'll have access to that information. And as Sam said earlier, if you would like to engage more in the Alliance, please check out their website and then feel free to follow up with Research America. You can follow up with me or Ellie, and we can forward that information to Sam. So thank you all. Really appreciate your time. We're going to just have a few very quick announcements. Please mark your calendars for our next Alliance member meeting. It's next Wednesday, the 28th at 3 Eastern. And we will be talking about the election landscape. And we'd also love your help in spreading the word about the Research America microgrant program. We fund groups of PhD and postdoc scientists who are interested in dialoguing with public officials to solve community-wide problems. And we have some terrific virtual formats that last year's class have developed and have been really successful in taking civic science virtual during the pandemic. So the deadline is November 9th. And just lastly, 2 weeks left to participate in Vote Science Strong. Thank you for all of those who have. You can easily send tweets, pro-science messages to candidates and your network, and we encourage you to jump on. Thank you all, and we look forward to seeing you next week.